Thank you, Jordan, and thank you for inviting me to give this very short talk, and I hope everyone can stick with me for the next 10 minutes. And so the work is really about a 3-year funded UKI work, really looking at the use of biocontrol agents for rice production in Africa. And the rice production has been increasing in Tanzania for many last decades, but the yield has been remaining very low compared with advanced rice production area. One of the 2 reasons they give to us is why for such low yield is one is disease control, primarily it's rice blast. The other is nutrient input. So we thought surely we can use kind of a beneficial microorganism for disease control as well as hopefully promote plant growth. So that's the rationale for this work. So effectively we say, can we combine the beneficial organism used as seed coating or as root dipping in transplant compared with foliar spray, foliar transplant to improve plant growth, control rice blast, then leading to yield increase. That's the whole rationale behind it. Because there's so much work in this project, I only presented one key aspect of the work, essentially how plant does respond to a beneficial organism. So what we show clearly, is if we apply same organism, Bacillus as root dipping or foliar spray post root dipping, and you have yield increase about 15 to 100%. In one particular year, applying both root dipping and foliar spray of same organism, you have additional effect. You can see on the right-hand graph 2021. So the increase the yield is more than 100%. Okay, so the question now is, furthermore, is if you combine the treat with 2 organisms applied different time, either Trichoderma in root dipping or Bacillus in foliar spray, or the other way around, you can clearly see on the graph you have additional increase compared with your same organism at 2 time points. So there's also a synergy effect between 2 organisms applying different time, not just same time, but different time. That's an interesting question. So the question we're asking is the yield increase due to the reduction in the rice blast only, which obviously is not the case. Reason being, if you see the graph on those points where you have shaded points, those are fungicide treated plot, have very low level of the rice blast, but the yield is about only a third or half of the highest possible yield. So essentially, we can clearly the yield increase is really primarily not due to the biocontrol effect or due to the reduction in rice blast. Okay. So then next question we ask is, okay, if that's the case, can we understand how plants react to the biocontrol or the application of the Bacillus and Trichoderma, and why the applying the organism at transplant as well as foliar spray have additive effect or even have synergy? So now, we did a lot of the RNA-seq work and use Trichoderma early, only treatment, look at the plant response following root dipping and foliar spray. Okay, so what we did is we root dipping and transplanting, then sample the leaf 4 weeks after root dipping. For foliar spray, we spray the leaf 3 weeks after transplanting, then sample 1 week after. So we at time sampling the rice leaf at similar age for between the root dipping treatment and also foliar spray. Very interesting. You can clearly say from the right-hand heat map, the leaf sample from root dipping are grouping together, clearly different from the leaf sample from foliar spray. It's incredibly clear. And also these are from 2 separate experiment replication over time. So really you can clearly say what happens is the plants respond to root dipping significantly different from foliar spray, although treated by the same organism. And moreover, as I said, the experiment was repeated in time. There was a clear difference between replicate experiments, simply because the different plants were used, or the season of time was used. Can clearly see the replicator one is a circle. The is open circle or closed circle. Then replicator two is open circle. Can clearly see they're separate so far away in kind of in that difference. So really, that also tells you the story. So whenever we do experiment on plants, rely on their response or RNA-seq, replicating over time is critically important, not just replicating at the same time. And given that so much results to talk about, and no time constraint, I'll give a very short summary. What is the result? So 1 week after the foliar spray, we still have about more than 3% of gene expression differ between treated and non-treated. And primarily those genes affected are grouped in two groups. Both are involved in plant response to heat and salt stress. And also the two groups were found with decreased expression really involve proteins for chloroplast and their interaction with the environment. And if you look at the root dipping, 4 weeks after root dipping, we still have more than 4% of gene expression differed. And for increased expression, these can be classified into 3 groups and include one group the same as leaf dipping. Unfortunately, not unfortunately, very interestingly, their directions are different. For the root dipping, the function appeared to be decreased, but for leaf seems to increase. So that really tells you is although the burst treatment led to yield increase, the plant response could differ significantly in certain biopathway, and that also possibly means the yield increase may be achieved through different mechanism. And if you look at the network of these genes, these affected genes, you can, for the foliar spray, we can class those affected genes into 5 modules. When you compare the treated with non-treated, you can clearly see that 2 groups of genes are actually different between treated and non-treated. That is the below the dotted line, the black dotted line. The first group is module related to the chloroplast, plasmid, and their interaction with their environment, or their movement, and also chlorophyll biosynthesis. Then second group of genes really relate to the calcium binding and plant passageway interaction. They're really affected by foliar spray compared with spray with long spray. But if you look at the root dipping, incredibly, there's far more gene module affected by root dipping, even 4 weeks after treatment. There are several groups of the modules with gene expression affected. We do not know their function at all. And there's few other groups of the gene affected. We do know they appear to be related to cell cycle, plant defense, ATP binding, chloroplast function, or etc. So clearly you can see is the root dipping affect the gene expression really much over wide, much more genes than foliar spray, and actually in different gene function. That may also explain why the additive effect of root dipping and the foliar spray of same organism on their positive effect on yield increase. So just to quick summarize, is essential we really say is root dipping somehow provide far more persistent extensive change than foliar spray. that also coincides roughly in critical experience in transplant, why you need apply beneficial treatment at transplant. For example, in apple, we demonstrate the long-term beneficial effect of, amending soil with beneficial microorganisms at the transplanting stage, have long-term effect benefit on apple growth and also on strawberry. Although there are similarity, one couple of the group of module similarly affected by the root dipping by foliar spraying, but also the clear indication, even for same group of genes, there could be differential upregulate by the root dipping and foliar spray, although their net effect on the yield is the same. It's the same increase yield, but the interest is that much more to the regulational mechanism, how yield increase achieved, rather than just look at the individual gene module affected. The large non-overlapping plant response between the root dipping and the spray treatment really explaining the additive effect of application, while the two applications work together, root dipping and foliar. Now we are really interested in trying to look at the synergy between using two organisms at two different times, how that actually even have more increased yield or synergy between the user same organisms. That's interesting. We're looking at the plant response level as well. So just to acknowledge the fundings from in the UK, our two African partner and in Kenya and Tanzania, and my colleague at NIAB support. Just to say, to clarify clearly, the yield increase was only observed, or the 50%, 100% yield increase was only observed in Tanzania in subsistence farming. But in Kenya, the increase is only about 10%, where the yield is already high. This is also clearly indicate the extent of the benefit offered by beneficial organism really is contact dependent, because more useful, more beneficial when there's a low input system. So I'll stop here. Thank you for your attention.